All right, we're now recording. Thank you very much, Jong, for agreeing to give a Build a Cell seminar. And you can get started.
Great. Thank you, Kate. Just to introduce briefly myself, I'm Jong Hyun Choi from Purdue. Again, like I said, we are a mechanical engineering group. So we do study engineering, but to build synthetic cells. So in particular, in this presentation, I'll be talking about the motility of synthetic cells by using DNA nanotechnology. So our skill sets are mostly in a DNA self-assembly to provide the motility and in particular programmable motility of synthetic cells. So that's what this talk is all about. So as in this community, we are all obviously interested in building synthetic cells that can resemble the structure and function of natural cells, biological cells. The motivation is obviously to elucidate the origin of life, as well as to understand the complex behaviors of synthetic cells, complex behaviors of natural cells. For example, in a synthesis of genetic molecules and cell division and growth and apoptosis and intracellular transport and signaling and membrane transport using membrane proteins and things like that. We, as a mechanical engineering group, we are interested in the motility, like I said. So we are using DNA molecules, engineered DNA molecules, to develop programmable motility for the synthetic cells. So when we look at the motion of biological cells, particularly those E. coli and other bacteria, so they show really interesting motion patterns and something like swimming and tumbling and things like that, which may be modeled as Lévy Flight or Brownian motions and things like that. Not only this in a diverse motion patterns, they also show really complex behaviors such as swarming and chemotaxis and things like that. So over the years, my computer is not responding, actually. Let me...
We can hear you.
Okay.
And we still see the slide.
Yeah, the slide is not moving, actually. I don't see... okay. Let me unshare briefly. Okay, hopefully it will respond again. Let me... Okay. My PowerPoint it's kind of frozen. They will restart. Just give me one or 2 minutes.
Maybe your computer has a jet lag too.
Hopefully it will behave.
Yeah.
Okay. Now let me reshare. Okay. Can you see the screen?
Yep, we see the slide.
Okay, great. So over the years, the lessons that we have learned is that simple molecular mechanisms can give rise to interesting diverse motion patterns and complex behaviors. So what we are all about is to use the DNA self-assembly and DNA engineering to provide those simple molecular mechanisms that we can take advantage of, right? So I assume that this community is familiar with DNA nanotechnology to some extent, but probably some have a lot of in-depth knowledge. Some others, have some maybe a little bit of knowledge. But anyway, so I will give you a really rudimentary introductions on DNA nanotechnology here. So obviously the idea of using DNA as engineering building blocks was proposed by Nassim a long time ago, like 40 years ago. He passed away a couple of years ago. But then the idea is really simple. Suppose that we have you have two double-stranded DNA in blue and red, and each one has a single-stranded overhang here. So if these two are complementary to each other, then obviously they will bind. This simple assembly using this simple assembly, then he proposed to construct a branched DNA junctions, and so that, in a larger scale, he could, perhaps he envisioned that he could construct a large macroscopic DNA crystals where he can actually place analyte molecules such as proteins and things like that, and for crystallography and or other applications. So since then, particularly last 25 years, there's been huge, progress in this area. People have constructed really interesting, diverse, complex structures such as polyhedra and teddy bears and smiley face and things like that. In addition to with this, constructing, interesting structures and designs, but also you can you can come up with a dynamic systems. You can play with dynamic systems. The most popular and most rudimentary, mechanism strategy is in a Tohn-Middleton displacement. Let me briefly, I'm sure, most of you are seeing this, and so I will be really brief. So imagine that we have two strands in blue and brown. So each one has two domains, 1 and 2, and 2 star and 3 stars. Since 2 and 2 stars are complement to each other, they will bind. But then now, if you present another strand in green that has 2 domains, 2 and 3, so what's going to happen is then 3 and 3 star will bind to each other because they are complementary to each other. And then what's going to happen then is these 2 in green and 2 in brown, they will compete against each other to take this 2 star. And ultimately, what's going to happen is, green strand will win out and win the competition, thereby displacing this brown strand. Obviously, this is a thermodynamically more stable, more favorable, but also there's a huge advantage in kinetics. Kinetically, this cohort gives you huge advantage. So using this toward strand displacement, a lot of groups and including us, design the dynamic systems. So in this talk, I will be talking to you, introduce 2 recent efforts, from our group. And, here, the first part, I will be talking about surface migration of synthetic cells by designing DNA walkers, or we call it also DNA motors or DNA walkers. And then later part, we designed and demonstrated a toy system, interesting toy system that can kind of shows collective behaviors using DNA signals. So the first system using DNA walkers. So obviously, intracellular trafficking by using protein motors, kinesin and dynein, walking along these microtubules and things like that. I mean, this has been a focus of has been interesting hot topic in biophysics for a number of years. Now we have a little better understanding. Then in DNA nanotechnology, also there's a huge, strong interest to develop, similar behaving, DNA. We call it DNA walkers or DNA motors. And so, we understand DNA, base pairing, and so that by using this DNA base pairing, we could design those kind of walking system for, transporting cargoes. So not only us, there have been several groups that have worked on this DNA walkers for a number of years. So I will briefly talk to you about the way we designed initially DNA walkers here. Our initial DNA walkers are using based on DNA enzymes. Let's just like 1023 and A17 DNA enzymes proposed by developed by Jerry Joyce's group. But not only that, we actually used other DNA enzymes as well. So basically, it has a catalytic core shown in green here at the center, and then it has 2 arms. So in red, upper arm and lower arm. And unlike this, protein motors, they use this, ATP hydrolysis as a chemical, converting chemical fuels for to move along these microtubules. And so instead we are using strands, DNA or RNA strands. In this case, we use actually chimeric DNA RNA oligonucleotides, where this catalytic core, appropriate conditions can cleave this pre-designed, pre-arranged these RNA points. So another difference between this protein motors and the DNA motors here is that ATP is floating in the around in the environment, but then those fewer strands for DNA walkers, these are planted on the track. So here we planted this RNA fewer strands on these single carbon nanotubes, just to using this microtubules and the cargos just like that. So we actually used the synthetic cargos in quantum dot fluorescent in the visible, and the carbon nanotubes, single carbon nanotubes fluorescent in the infrared, near infrared. So with that, so we are using this single carbon nanotubes fluorescence as well as the quantum dot fluorescence to visualize the track as well as the DNA walkers. So what happens, the way we our system works is that under certain concentrations of magnesiums and all other cations, this catalytic core can cleave the prearranged RNA point, and they're leaving this two fragments in P1 and P2, and over time, and this P1 will dissociate, thermally dissociate, and move float away. That now our upper arm is free so that it can actually graft the next available RNA, and then the lower arm will follow up. So you can kind of think of it as remote toward strand displacement. So this is really describing a single turnover step, right, in a walking cycle, so that by repeating this cycle, our walker can move autonomously without any human interventions and moving forward. Because you are cutting, we are using enzymatic reactions to drive this movement, so that this is a bond bridge mechanism. So it cannot move backwards; it has to move forward. So that's another difference. Okay. So to monitor our walker system, we had to kind of design our microscope system where we actually embed our DNA walker system on this microfluidic chamber, and then we use two different lasers, two different cameras. Here we use a indium gallium arsenide to monitor the single carbon nanotubes, and for visible quantum fluorescence, you can simply look at the visible luminescence using a EMCCDs, but carbon nanotubes, you have to use a special cameras such as indium gallium arsenide. But anyway, so this is a typical immobilized single carbon nanotubes and quantum dot. Obviously, this is a you see a optical diffraction. So to overcome that, we use a localization method like many others. So by using a Gaussian fitting, so that you can actually monitor quantum dot locations along this single carbon nanotubes. But then this, visible quantum dot imaging, sub-diffraction imaging is kind of straightforward. But then carbon nanotubes, this is a little bit of a challenge because of its 1D nature, right? You can think of it as a concatenation of spherical flow force, one-dimensional concatenation of spherical flow force. But also important thing is that carbon nanotubes, fluorescent, they don't really photo bleach, they don't bleach and they don't blink. But instead, one thing that we do know, obviously I came from the carbon nanotube community, but one thing that we know is that whether it's DNA or RNA wrapped carbon nanotubes, then the surface coverage is not as tight as typical detergent such as SDS. So at lower pH, something like 4 or 5 or something like that, then what could happen is then if you take multiple images and then subtract one from another, then you can kind of isolate the emission centers. Then you can apply this sub-diffraction superposition imaging methods, localization imaging to on this isolated imaging centers, so that you can reconstruct the sub-diffraction image of superposition image of carbon nanotubes, and also you can kind of look at the quantum dot, so that you can actually track the DNA. So this is a kind of typical measurement, so that for immobilized carbon nanotubes here, still it looks kind of large, but it's really 1 nanometer in diameter, and then this quantum dot is moving in one direction by using DNA walkers. So using that, this measurement platforms, we were able to gather a large quantity of kinetic data so that we can actually study their how to design them and to have a better, really highest performance possible. So since then, using, now we know a little better about how to design DNA walkers. So our approach is that using these DNA walkers to attach and decorate, functionalize these, vesicles, so that we can actually take advantage of this DNA movement, DNA programmed migration movement on this synthetic vesicles. So the way we constructed our synthetic cells is that using a couple of different phospholipids and P and PC, and then we also use a click chemistry to attach DNA walkers, designed the DNA walkers onto lipids, and then also for visualizations we use a fluorescent probes, and then so we do the self-assembly to form a lipid bilayers and synthetic vesicles. So synthetic vesicles in this case, actually, as we'll see in the next slides, and it's fairly small. So small unilaminar vesicles expressed with functionalized with DNA walkers. And then our idea is that not just moving, carbon nano to one-dimensional track. So we are not really. I mean, that's how we actually, gain the knowledge of how to design the DNA walkers. Now we are interested in the migration, 2D surface migration of these synthetic vesicles, small vesicles. So as you see, TEM and DLS measurement actually indicates that the average diameter is roughly 200 nanometers, although there's some distributions, about 200 nanometers. So the way we designed was that vesicle has a number of DNA walkers. In this case, shown in red is expressed, but it's only showing in only one in this cartoon. But then the surface is functional with RNA, and then we're going to use a DNA walker and then RNA. In this case, we are not really using DNA enzyme anymore, because DNA enzyme is actually fairly slow. So we are now using an enzyme. We explored RNase and XO3 and many others, but then RNA. What we learned is that RNase gives you the best consistency. So we are going with RNase from now on. So the enzymatic reaction, digestion moves this DNA vesicles, so that actually, this is also a bond bridge mechanism, but then you're talking about in 2D in a plane, so that actually you want to move away from your own current spot. So that's going to be your next movement, right? So this is, you can think of it as a self-avoiding fashion, self-avoiding walk, random walk, right? So if you run some, stochastics, Brownian dynamic simulations, as a function of time, this is actually a Fourier number. Fourier number is defined as a diffusion coefficient and time and particle radius. So really, physically speaking, this is gives you the idea of physical dimensionless time. So if you plot mean square displacement, obviously theory gives you the self-avoiding motion. It goes sits in between the diffusion and drift ballistic motions, somewhere in between. But then our simulation actually gives you the idea that yes, it actually works. Starting initial short beginning time is behaving like diffusion, but then it transitions to the self-avoiding motion. So, however, that's the theory. But then keep in mind that this self-avoiding, this stochastic theory actually is assuming a constant velocity. But then what we see in the experiment, it's not exactly. We see a different types of behaviors, random walks, and sometimes we see ballistic shown in red, and sometimes we also see Lévy flight, long straight Lévy flight, super diffusive Lévy flight, but also as well as self-avoiding motions, and also finally diffusive motions as well. So if you look at individual, motion behaviors, for ballistic motions, we see some, hopping movement. So that there is a, step time and velocity it varies, has a broad distributions. And Lévy flight is a long stride. Lévy flight is far more consistent, but still, it has some variations in their time step and step time and velocities. So self-avoiding motions is more consistent, but also not only that, but also it has a more narrow distributions in velocity as well as step time. And obviously, Brownian motions is kind of confined within the area, right? So using this, by monitoring these different types of motion behaviors of vesicles, so we actually wanted to actually have a better understanding and better modeling, having a random walk model with random velocities, so variable velocities, variable step time, so that we could actually describe different of our experimental observations. So the plot, according to our random walk model, what we got is that on the y-axis we got velocity distributions, and on the x-axis step time distributions, and in pink to describe this theoretical ballistic, and the super diffusive in orange color. So super diffusive includes Lévy flight as well as self-avoiding motions, and gray shows the diffusive motions. So if we plot our ballistic observations, experimental data, it actually falls into the ballistic profiles, and then Lévy flight as well as self-avoiding motions. These two constitutes a super diffusive behaviors, and obviously the diffusive motions. So with this model, we have a little better understanding and so how to provide a different types of motion behaviors to these our small vesicles. So with that, we subsequently performed a mechanistic study by exploring different parameters, something like the length of the DNA walkers and the DNA designs, or environmental factors such as the temperatures or magnesium concentrations, so salt concentrations and buffer conditions and things like that, and part in the cargo size, right? So we ran through a number of parameters to develop a more mechanistic understanding. So skipping all those detailed studies, now we know how to really focus on how to demonstrate more single walking mode, predominantly single walking mode. For example, self-avoiding motion. This is a self-avoiding motions of a small vesicle, individual vesicles. So it can travel up to something like 20 microns, and walking time is more than it's really processive, more than migration is more than 10 hours. The velocity wise is still fairly slow, 2 nanometers per second. Obviously there are different ways. I mean, if you design more ballistic motions, then you can go much faster, but then it's not going to be really as persistent, as consistent. So we prefer to have sacrifice velocity a little bit, but then we have a more processivity and more consistency in our migration ability. So with that, what we did was we wanted to actually explore more interesting cellular behaviors, such as chemotaxis. Obviously, I assume all our this audience seeing this really this video, neutrophil chasing bacteria, this chemotaxis. So we wanted to actually come up with a system, vesicle system, such that we can do similar chemotaxis. We call it a synthetic chemotaxis. So to do that, we actually designed 2 species of vesicles. We call it lead and follow. Lead, just for our cartoon, we use a red, color coding in red, pseudocolor red, and then green for follow. But then important thing is that, as you understand, unlike typical chemotaxis that you use a concentrations of chemoattractant or chemorepellent for information transfer, here we use a fuels attached on the surface to store the information about the trajectory of the lead vesicles, right? So our design, fuel design, is shown here schematically. So it has a hairpin strands, actually hairpin conformation. So that actually where you store the information of the lead vesicles. So imagine lead vesicle binds with the fuel, on the surface, then what's going to happen is that it will open up the hairpin core, and then the enzyme will move the lead vesicle away. And then subsequently, follow vesicle shown in green will come and bind with this opened active fuel, and then it will move. But then important thing is this movement, enzymatic powered movement, will actually have a huge bias. So this follow vesicle will experience bias, strong bias to track the trajectory of the lead vesicle. So that's the idea. I will show you how we actually achieve that. So the follow vesicles, without, if it is the strand, fewer strands is, initially bound or something like this with hairpin conformation, then obviously the follow vesicle can bind, associate with its fewer. However, it's going to be too short, and then it's too short for cleavage, and so it's going to be, you may move a little bit, but it's going to be fairly more or less, it's going to be inactive. But then if it is opened after this removal of this fewer, open the hairpin core, then the follow vesicle will be have an active fewer, and then it will move as designed. So this is the trajectory, trajectory of the, this follow vesicles more freely. Not only the trajectories, but also when it comes to the speed and velocity. If it is inactive, then it's fairly, it doesn't really move well, but then with active fuel, then follow vesicle can move well, as designed, between something like 1 nanometer per second or something like that at an average velocity. So, before we, perform the experiments, we ran the simulation whether this is possible or not. And so, with just like we had a stochastic simulations as a function of Fourier number. So initial lead vesicles move freely in random walk, but then after that we seeded a follow vesicles with some strong bias, so that the green follow vesicle will only stay along this course, the trajectory of the lead, right? If it is seeded somewhere else, it may move slightly a little bit, but then its movement is going to be really limited. So to characterize these different path tracking, correlated path tracking, and negative control, we came up with two analysis platforms. One, it's called colocalization coefficient that measures spatial correlations. And the other is angular metric for shape similarity that measures temporal correlation. So without going into all the details, let me show you how it shows up. So this is a colocalization coefficient in correlated path tracking and negative control. So this is a position index of the lead vesicle, and on the x-axis, this is a position index of follow vesicles. As you see, when there's a correlation, when there's a chasing of following the tracking of the lead vesicles by the follow vesicles, then you see a strong really high intensity along this diagonal. But then in contrast, if there's seeded somewhere else, then follow vesicle doesn't do anything. You cannot really track the trajectory of the lead, then there's nothing happens. When it comes to AMSS and the temporal correlations as well, and so the path tracking, if there's correlated, then it goes from 0 to unity. This is normalized, so it goes from 0 to unity as a function of non-dimensional time. Dimensional is time Fourier number, but then negative control doesn't do anything here. So now I'll show you this is our experimental observation. So that initially the lead vesicle in red moves freely by random walk on the glass surface, but then the follow vesicle in green, follow vesicle actually seated somewhere close to the lead vesicle, but then quickly tracks on the trajectory of the lead, and then follows the trajectory of the lead vesicle, the red, right? So what happened here is that this follow vesicle, the blue and green, can move exactly the same, taking the same direction or completely opposite directions. So if you really think about it, this is we are applying the bias, and the information is really embedded on the track surface, right? So the follow vesicles, when it comes in, then the initial symmetry breaking is, it's random, but once you move in one direction, whether it's following the same directions or opposite directions, then you have to follow that directions. So that's exactly what we see. So if you plot this, in our case, it goes the opposite directions. In other cases, we see that same directions too, but then in terms of position index, so that's why it's actually flipped. But this is the lead and this is the follow. As you see, you can you imagine the high intensities along this diagonal. I mean, obviously it's uncorrelated. Obviously, there's complete dark, as you can imagine. So time correlations, temporal correlations, AMSS, and also if it is correlated in a time path tracking, goes from 0 to unity, but then negative controls, there's just a noise as a function of Frey number. So here we are fairly happy to see this this toy system chasing each other of these vesicles. But then where we really want to head it to is one more thing. So before I forget, so in terms of the speed-wise, the follow vesicle is shorter than long vesicle, the lead vesicle, so that it's faster. So you can actually even design a chemokinesis such that follow vesicle can track the lead vesicles, and then just like neutrophil engulfing bacteria, you can actually design such a similar animal predatory system using vesicles. The next topic that we wanted to show you is that, again, so far our vesicle migration system was really between the two vesicles, one in red and the other in green, right? Only between the two. It doesn't necessarily have to be the only two, but that's what we've been mostly seeing. So, but what we wanted to do was more like a collective behaviors. In this toy system, I will show you, we were interested in building a toy system that can actually, you can work with multiple different vesicles. So to do that, what we had to develop was a information transfer or communications between signaling pathways, signaling mechanism between vesicles. So what we did was we constructed DNA Origami as a membrane pore, okay, and then we are going to use this DNA as a signaler. So and also what we also wanted to do was using a DNA Origami as a membrane pore, just like that, so that we can actually have a DNA Origami here, so that we know how to control the in and out, what can enter, what not to enter, and things like that into a giant vesicles, giant unilaminar vesicles. And then inside, obviously, also what we wanted to do was to have a ability to transduce a signal, right, DNA signal, so that we actually embedded a XO3. Plus, at the same time, because we are using a DNA Origami channel, it actually has a fairly large diameter. So we were kind of afraid that XO3 enzymes might, diffuse out of these giant vesicles, so that we actually functionalized XO3 enzymes onto these nanoparticles, so that they won't be able to move out. So this is the giant vesicles that schematic cartoon that we constructed giant vesicles, and we have multiple nanoparticles modified with XO3 enzymes, and also on the surface it has a DNA, single-stranded DNA expressed on the surface, and the DNA Origami membrane pores. And so our idea is that our rudimentary, really simple, cartoonish idea here is that, hey, if you have it giant vesicles, but then you can also have a small vesicle, just like SUV that we have, played with, presented just a few minutes ago. Then perhaps, this small vesicle may have also different, DNA, set of different DNA strands, single-stranded DNA, such that, if you have a DNA signaler, hairpin signaler, so hairpin alone as a hairpin structures, it may not do anything. But then if it enters the large vesicles, and then it may be digested by this XO3 and then it can go out and then binds with, has two domains. One domain binds with this green DNA on the giant vesicles. The other domain binds with blue on these small vesicles, such that this DNA signal will induce a aggregation of small vesicles onto giant vesicles. Okay, but also at the same time, using this strand displacement, you can actually remove this, linker strands, the DNA signal, such that you can actually get rid of all the bound SUVs. So that's the toy system we designed. So to be able to have a communications between large, vesicles, giant vesicles, we need a DNA origami as a transmembrane pores. So we are not the only group who constructed and demonstrated these DNA origami channels, and Fritz Seymour and Uli Kaiser and others too. So basically, our design has a DNA origami tubule, has about 30 nanometers in diameter, so fairly large, because that's what we wanted, and we wanted to actually have it in proteins and DNA in and out, so that that's what we wanted. Also at the same time, to insert our DNA origami, we actually modified DNA origami surface with cholesterol moiety, so that we can actually insert this DNA origami channels into the giant vesicles during this its self-assembly. At the same time, we actually have some linker strands on the on the tip of this origami channel, and also we wanted to have design a cap so that we can actually cover, and so that we can actually control what's in and out. So again, here is a linker strand, so that these two will bind, these two sets will bind, so that we can actually form this kind of structures. So on the left, with the DNA origami, actually we modified the DNA origami structures channels with some fluorophores, so that it shows up here. Without the DNA origami channels, obviously there's nothing in there, obviously. So it's just that this is a fluorescence image and the graphite image, so that the giant vesicle is there, but then you don't see any DNA origami pores. Here we do have pores. Okay, so you can kind of control what's in and what's out. So first, what we did was testing the pores, so that with the pore, obviously you can actually put the GFP and Psi5. Actually, this is Psi5 modified with DNA. About, if I remember correctly, something like 50 or 55 nucleotides, so kind of oligonucleotides modified with Psi5, so that they can enter or move out, diffuse out. So that's the freely they can all in and out the giant vesicles through these DNA origami channels. But then without the channel, obviously they cannot enter, obviously. But also with the cab, you can kind of control their transport, right? So initially we constructed. Initially we embedded GFP and Psi5 DNA within the giant vesicles, and then after some time later, we removed the cap, the lead of the DNA origami cap, DNA origami channels, so that then by using a set of releaser strands, so that we can actually remove the lead and the cap of these DNA origami pores, so that since then, shortly after, a few minutes later, the GFP is inside and a Psi5 DNA inside will move away, will diffuse out. Actually, one thing that we found was that by fitting this curve fit, it's about kinetics is about 7 minutes or so. But then interestingly, it doesn't the fluorescence it doesn't exactly goes all the way down to 0, but it's always stays something like between 30 and 40%. So that was quite interesting. Before we actually demonstrated the full-fledged toy system as we designed, as I mentioned, but we had it in a smaller, simpler version. So without having any DNA origami pores, we just had it, giant vesicles immobilized on the surface, and then we used a linker strands and releaser strands to go between the aggregated and assembled and disassembled structures. So as you see, this is a this is pseudo-colored giant vesicles under the microscope, and then once you have a linkers and small vesicles actually has only small vesicles have the fluorescence probes, not the giant vesicles. Just fine vesicles, obviously you can look at it under the bright field image, but then once the you present the linkers, obviously of course they are bound, but then if you put the if you introduce a set of releasers, then you can actually take out all these bound small vesicles from the giant vesicles, right? So that you can repeat that process. Although it doesn't go all the way completely zero, but I mean, you get the idea that, perhaps maybe we could have, added more higher concentrations, I mean, DNA, the releasers, set of releasers, but still, at least we know it works. Although it's not perfect though. Again, if you use a kind of random strands instead of linkers, of course, it doesn't do anything, right? So that's what happens. So now this is exactly how we designed initially. So initial design, as you see, we have a giant vesicles with DNA origami as a pore. Initially, it was capped. The lid was blocked any DNA origami pores. The inside the giant vesicles, we have XO3 modified with this polystyrene nanoparticles floating inside the giant vesicles. As you see, this yellow, if you look at it as a movie, then actually they are moving around, they are floating inside of vesicles. But then once we introduce the cap releaser strands, so DNA releasers, so that we can actually open the cap here. We open the cap here, and then we introduce the hairpin. So the hairpin alone, it doesn't do anything, as we'll show you in a minute. But then with the hairpin, that can enter the large vesicles and then get digested and then move out. It can induce the aggregation of small vesicles so that you can see strong luminescent fluorescence from these small vesicles. And once you provide the releasers, of course, you can actually get rid of it, although it doesn't go all the way down. But then there's a significant drop in the fluorescence. And then you can actually repeat this process. So of course, as a negative control, if you use you can open the cap, but then if you use a random strand instead of hairpin, then it doesn't do anything. Also, if you don't open the cap, and even with the hairpin, not including here, but then there's no change in the fluorescence at all. So with that, we demonstrated this toy system for this aggregate DNA programmable aggregation of different vesicles, small vesicles onto the large vesicles, giant vesicles. So with that, where we are headed from here is that we're more interested in now a swarm behaviors. That's where we really want to go, right? And swarm behaviors of this synthetic vesicular systems. That's the effort that we are putting at the moment. Okay, I'd like to thank my students who did all the work and funding agencies, and thank you for your attention.
Thank you so much. That was a great talk. There are some questions in chat. Can you see chat? If you could read the question out loud before answering, that's going to be great for the recording.
Right. So let me jump up to the different message here. Okay. Let's see. All right. Oh yeah, mechanism dependence. Obviously, yes. If you look at the DNA nanotechnology, DNA behaviors are really based on you need a fair amount of magnesium. So most of the time, okay, not all, but we actually initially we studied it particularly for DNA walkers. So we actually played with concentration dependence of DNA motors. So there's a strong behaviors. You gotta have at least certain amount, like 10, 20-ish. So, you know, if not, then if you have zero magnesium, then it's not gonna really work. So you at least you want to have some. So 10, 20, you know, obviously we need. So that's the important, you know, ingredient that we may have to maintain. So obviously you can actually play with it a little bit more. I mean, like you can have a little higher concentration or less concentration, but you know, at some point we may have to have some level of magnesium, or you know, at least you know, some cations. So that's the first question. The next question that I see from Tanner. Composition matters. You know, actually, we haven't really dealt with a lot. I mean, we just used you know, different components on the different PE and PC concentrations, and it, you know, at first glance it didn't really matter too much, but we didn't really use pegylated liposomes. I mean, but if I think about it, I mean, why not? So pegylated system, I mean, the reason why we didn't really use the pegylated, liposomes was, we use it to passivate our surface, because again, we need to place our fewer strands on this, cover slip, glass cover slip, right? So that if there's some, defects on the surface, we easily get stuck. So surface passivation is very important for us. So we use actually, the PEG to cover up. So that's why we didn't really use pegylated liposomes. But I mean, if I imagine, I mean, it might work. So we haven't, but personally, we haven't really tried pegylated liposomes, but I assume it will work as well. Hopefully I answered the question. The next question from Herbert, could you use pH for motility control too? Yeah, I mean, like that's another that's very important, pH. So that's something that we are really trying to demonstrate, particularly pH. I mean, with pH, the way we wanted to use a pH is that we want to use a triplex DNA and things like, so that we can actually use this pH as a another control factor, control mechanisms, right? So all the things that we presented now is that mostly it's just a single pH, whether it's a sometimes 7.4, sometimes a little bit less, for particularly with the carbon nanotube. Superposition of carbon nanotube fluorescence, there's a little bit less, like something like 5 or 5.5. That was the pH back then. But then, so we want to use this pH by using triplex, DNA, so that we can kind of use as a control. Imagine, you can kind of imagine, with pH, you can kind of induce a, reversible aggregation at lower pH, and then using a higher pH to reverse that course and things like this. So that's something that we are very interested in. That's something that we are working on currently. So hopefully, we will be able to report that, soon. The next question I have is from Harvey. So not the really research question, but the tracking graph looks like the northern lights. Yes, I mean it's so we have a series of measurements, and so I mean some we reported in a paper SI, but also there are a bunch of others that we haven't really reported. So that's a it's a really a random walk, right? So that's a that we didn't really control in terms of, particularly in terms of the first lead vesicle random walks. So the next question from Lucia, when you cycle through addition of SUVs and small unilaminar vesicles and release of strands, do more and more residual SUVs accumulate, or does only the first cycles? Oh yes, I mean that's a really important question. So we didn't really see a huge accumulation over time. I mean, we did. I mean, we did only a few cycles, right? I mean, because adding and waiting for the binding and all that. So it's a kind of time-consuming experiment. So we couldn't. I mean, my students kind of refused to do multiple cycles, right? But all the cycles that we have done, number of cycles, several cycles, like we've done, not exactly over. We didn't really go over 10, right? Because it was too long, but several cycles. I mean, the most signal change was the due to the non-specific absorptions, probably first cycles, and then didn't really see much after that. I mean, a little bit, yes, but mostly came from the first cycle actually. But then that once, as you've seen here, let me go here. I mean, so as you see, first cycles and obviously there's a little bit of changes, but then even we go a little bit longer, like 7, 8 cycles, still it was fairly similar, right? But one thing, again, as I mentioned, we didn't really use high concentrations of releasers. I mean, I'm not sure. So again, it will make hopefully it will make some more difference, but then perhaps next time we could do a little better. Yes. So let me move on to the next question. So how are the kinetics of the aggregation and the reverse process? Yes. Yes, this is a really good question. And I mean, obviously, this is something that we couldn't study much. So I mean, the bottleneck is the division of the hairpins. And absolutely, yes. So we have to really rely on the diffusion, right? I mean, the hairpin signal has to get in. We know it works, the scheme works, but then it's the bottleneck is the diffusion, right? So it's all the entering of this hairpin signal into the giant vesicle and have a signal transduction, digestion by the enzymes, and then the processed, this, hairpin signal will have to go out. So it's really depends upon the, kinetics and concentrations and things like that. So yes, but then releasers, I mean, that's fairly fast. The releaser, toward mid-exchange displacement alone, that's actually, as long as you have a high concentrations. We didn't really, I mean, I could be wrong, but if I remember, we used something like 20x concentration, so that that actually worked fairly quick. But the aggregation, we have to wait. We gave enough so that even though it shows that straight up, but it's not exactly straight up. Between the experiments, we have to wash it and all that. So it was a slow process actually. Okay, so let me go next. The final question is, have you tried using cholesterol labeled walkers? Oh, that's a good question. And based on your talk, I assume conjugation walkers directly to the lipid walk better than cholesterol labeled walkers. So, yeah, I mean, so we didn't really use a, I mean, we could have, right? I mean, but we just used that, we just used the, the click chemistry, right? I mean, so that, I mean, I guess we have to go all the way down up. But the question is, I mean, our students are mostly in mechanical engineers with their background, so we didn't really use it as cholesterol. We just used it as simple as possible. I mean, I guess you can call it them cholesterol labeled walkers are even simpler, but I don't know. But we just used the click chemistry, and it worked out fairly well. So we didn't really explore more, but theoretically it should work too. But did I hopefully I answered all the questions.
Looks like you did. I was really curious about the pH question too. That
yeah,
that was really cool. All right. If there are no more questions, thank you so much. And
thank you for your time and I mean, me coming from different backgrounds and different skill sets, and I mean, one of the things that I, really found it impressive and really fun is that, this community is really collaborative and exchange ideas, and I mean, like in any other, communities. But again, I came from graphene and carbon nanotube community, so that's why you see. Why do you use even carbon nanotubes as a track, right? I mean, you could use DNA Origami and others, but I mean, it is partly because I came from different community. But then, really, it's a great community to exchange ideas and skill sets and promote collaborations. I truly appreciate all of you guys in the community and the effort. Thank you.
And we appreciate your work. Thank you so much. And I agree that this is a really friendly community. All right, thanks everyone, and have a great rest of your day. Bye.
Okay, bye.